Hello. So we're going to get started. So thanks so much for coming to this session on the climate drivers of infectious disease. We have 8 amazing presentations. So they're 15 minutes each, including time for questions. We also want to let you know that this is being live recorded, just the audio and the visuals as well. So you'll be able to access it on demand after the talk's done. So our first speaker is our invited speaker, Professor Jeff Shaman from Columbia University. And he's going to be talking on the use of ambient humidity conditions to improve influenza forecasts.
Grand entry there. So I actually have a bunch of information that I have to go through to contextualize this. The first is to do with the basic structure of influenza transmission dynamics. So what you're seeing here is an average of 31 years of excess pneumonia and influenza mortality, which is a good indicator of influenza incidence in the broader population. It's shown for New York State and California states from 1972 to 2002. It's a climatology of influenza incidence in essence, and what it shows you is something that for those of us who have lived in a temperate region of the world we intuitively know, and that's that you get influenza in the wintertime. Now there are a number of possible hypotheses that try to explain why we actually get that. This has been something that's been observed for over 2,500 years, and we really haven't nailed it down at this point. You can put them into 3 groups. The first has to do with changes in host immune function. That might be linked to the environment in and of itself. During the wintertime, the sun's at a lower angle in the sky and day length is shorter. You have less melatonin and vitamin D production, and as a consequence, you have suppressed immune function, which may make you more susceptible to infection. And furthermore, once infected, you may shed more of the virus as you're not able to fight it off as effectively, so you're more contagious. The second group of hypotheses has to do with crowding in indoor conditions during the wintertime when people spend more time indoors, in theory. So there you again have increased person-to-person contact as a result of people spending time indoors when it's colder, and as a consequence you have an increase in the transmission of the pathogen. The last has to do with external conditions, and they may have to do with ambient conditions in the environment, humidity, temperature, etc. So in 2007, late 2007, there was a paper published out of Peter Palaze's group, who's a virologist at Mount Sinai in New York. It was led by Anise Lowen, who's now at Emory, and they ran a chamber experiment using guinea pigs, which they found to be a very good animal model for studying the transmission of human influenza. What they did is they ran 20 experiments with a chamber in which they took 8 guinea pigs at a time. They infected 4 of them with influenza, and the other 4 were to be susceptible. They structured the chamber just as you see here, with infected and exposed, and a slight breeze going from infected towards exposed. Guinea pigs are in separate cages within this chamber. So the transmission has to be airborne, and after 72 hours at fixed relative humidity and temperature conditions, which they varied from each of the 20 experiments, they then measured how many of the guinea pigs actually came down with flu. What they found were marginally statistically significant effects in which colder temperatures and lower relative humidity conditions seemed to favor transmission. So I read this paper and I wondered why they were looking at relative humidity. Relative humidity is very important for certain biological phenomena, molds, gram-negative bacteria, etc. But why they were using something while varying the temperature didn't quite make sense, because you monkey around with the temperature, you monkey around with relative humidity without actually changing the humidity content. So why not use a mass-based measure of humidity? So what we did is we took their data and we rejiggered it. We used just Clausius-Clapeyron equation to calculate the absolute humidity in those chambers. And when we did so, and included in addition some additional data they published at high temperatures, what we found was a more striking statistically significant relationship, particularly shown on the right, that is a little bit nonlinear between the number of guinea pigs that are getting infected with flu and the conditions inside. Specifically, we found that with drier absolute humidity conditions, there was an increased likelihood that you were going to find more guinea pigs infected with influenza. We looked through a number of the hypotheses that it put forward. Some had to do with cloud microphysics, droplet formation, and whether droplets settle out or evaporate and are aerosolized. Those didn't really pan out. They had another hypothesis in this paper that it was related to the survival of the virus that was somehow modulated by ambient humidity conditions. So we went combing through the literature again, and there are a number of papers going back all the way to the late 1930s in which they would take influenza, culture it, atomize it, stick it in a canister for a period of time, and then sample it to see how long the virus survived in different conditions. One in particular from 1961 by Harper actually was kind enough to publish the data in their paper and to tested out for both temperature and relative humidity. None of the papers had actually looked at absolute humidity. So we did the same exercise here, and what you're seeing are dots that are showing on the left, relative humidity on the top, temperature in the middle, and vapor pressure, measure of absolute humidity on the bottom. All three of them go from left to right, from lower to higher, and then you're seeing the percentage of the virus that was viable after 1 hour, 6 hours, and 23 hours. Those are in the different colors right there. And if you see, what you see is something similar to the guinea pig experiment. Basically, the virus remains more viable at lower relative humidity conditions, at colder temperatures, but then when you get to vapor pressure, you see a very, very clean relationship. And as a matter of fact, if you do a simple log linear regression, 90% of the variance in influenza virus survival is explained by absolute humidity, this one marker. So that seemed to be a bit of a smoking gun. Furthermore, whoops, gotta hit the right keyboard. Furthermore, if you look at the seasonality of absolute humidity, it's the same indoors and outdoors. We modulate the temperature by heating and cooling it, but we don't do much with the humidity content of the air. So basically, we have very low humidity conditions, absolute humidity conditions in the wintertime, and they're very high in the summertime. As a consequence of it varying this way both indoors and outdoors, it's in phase with the relationship. It's minimal both indoors and outdoors in the wintertime, and thus favorable for the survival of influenza and the transmission of influenza. So the next thing we asked is, could we use these observed conditions to actually simulate and model influenza transmission? What we did is we built a simple compartmental model. It's really a two-variable nonlinear oscillator forced by observed absolute humidity conditions. And we applied it at 5 states with dissimilar humidity conditions: subtropical Florida, desert Arizona, funny Pacific Northwest Washington, New York, and Illinois states. We took that nonlinear relationship that you see there between the viability of the virus and humidity conditions, now switching to specific humidity because it's more routinely measured, and we had a functional relationship that actually imposed within the model a variation of the survival, the viability, really the transmissibility of the virus in a similar fashion. We then would run it for 31 years with 2 influenza viruses cycling through, and we'd collapse it and get a climatological output of what it showed in terms of the seasonal cycle of influenza. And we found, if you look at the plots there, those are the results for the 5 states. The red dashed line is the observed excess influenza and pneumonia mortality, similar to what I showed you on that first plot for the 5 states. The blue line is the best fit line, and the green lines are the 2nd through 10th best fit lines. The important thing is that they all converge to the same parameter space. In other words, the simulations that did well and fit this well all moved to the same suite of parameters controlling the dynamics of transmission within the model. Now, this is very important because in the model like this, the parameters are dictating biological processes. And if we had to draw on a different set of biological processes to make the fit work in New York versus Arizona versus Florida, we're in essence calling on different biology and in essence insisting that there's a different flu pathogen out there, which we know there isn't. So the fact that it converged to the same set of parameters was very valuable. We then actually could cross-validate it by applying those best fit models to the other states that we haven't shown here, and we were able to make fits of similar quality. So the next question we asked is, can we actually predict individual outbreaks? Could we take a humidity force model of influenza, these simple models we have, and predict the variable outbreaks of flu that you see there? Those are time series from New York City. Ignore the yellow, which is the second wave of the pandemic, but the other ones show that there's real discrepancies between the magnitude and the timing of individual influenza outbreaks. The answer to the question is a simple no. The model by itself cannot actually make these predictions. It's not sufficient to just have a simple model like this with its nonlinear dynamics and expect to make a credible prediction. However, we can mimic the strategies that have been used in numerical weather prediction and bring on board data assimilation or Bayesian inference methods and optimize the model to an individual outbreak as it has thus far been observed, using observations of the system that are available in real time and generate forecasts. And that's exactly what we did. We took our simple 2-variable nonlinear oscillator, we combined it with estimates of influenza illness, this time it's called ILI+. We used a data assimilation algorithm just like they use in numerical weather prediction. Here we use the ensemble adjustment Kalman filter on this simple model. And the whole point of doing this is to try to account for error. There's error throughout the model structure, which makes it problematic for actually making predictions. The model itself is misspecified, there's error in the observations, and there's error in initial conditions. Left to its own devices, if in state space this is the true outcome, our model might do something like this. To make it look like an outbreak, it might be like this, where the true outcome is in black and our forecast may be in red. We don't want it to do that. We want it to do something a little better. And that's particularly boneheaded if those are weeks on the bottom, and that's week 15. We have information showing that there's been an uptick in activity, and we don't account for that in the model. We want the model trained on what's happened thus far so we can make a credible prediction. We do this in ensemble form. When it works, it looks something like this. This is a real-time forecast that we generated for Salt Lake City in the 2012-2013 season. The black X's are observations we had in hand at the time. The red X's are observations we did not have. So you can see that it captures the timing of the peak, the overall duration, and the attack rate, and we're predicting the peak of the event 5 weeks in advance. Furthermore, we actually can do calibrated forecasts. We can open up each one of those gray lines that you saw there, which is actually the mean of an ensemble of predictions, and we can look at the uncertainty in it. We can use that to calibrate the forecast. We don't just say that flu peaks in 5 weeks, we can say there's a 70% chance that flu will peak in 5 weeks versus a 20% chance that flu will peak in 5 weeks. Very analogous to a precipitation forecast, but it gives you actionable information by giving you a clue as to the certainty of the forecasts. So we've been doing this operationally now for 5 years. Here's last week's forecast for New Orleans. It's predicted to peak in about 4 weeks in terms of flu. It's an early season H3N2, and I would encourage all of you to get vaccinated if you haven't. We next asked this question. Within our model, we have humidity forcing. That's the basic core model that we use. We wanted to then address the question of whether or not inclusion of humidity forcing actually improved the forecasts. Now, when we use the humidity forcing, we're actually using a climatology. We don't know 3 months from now what humidity is going to look like. There's a lot of synoptic variability. We're never going to be able to account for that. So we tried out, in retrospect, 4 different ways of addressing humidity forcing. The first was to use what we've been using operationally, which is just a daily climatology of absolute humidity conditions for the locality we're making the prediction for. We did another one where we used the observed absolute humidity. It's retrospective, so we can cheat and know what actually will happen in the future. It's not something we could do in real time. The third one, we actually did a kludge of it, where we train it with the observed up to the point of forecast, and then use climatology in the future, which is something we could do in real time. And then the fourth, we excluded humidity forcing. We didn't put it in the model at all, and we just ran a simple model of the dynamics of transmission. What you can see here is error, or the accuracy of the peak intensity forecasts and the peak week forecasts, which is the timing of it. It's showing you the fraction of the forecasts that are accurate based on how far in the future they're predicting the outcome will be, when the peak will be. What you should really note is that particularly for peak intensity, there's a big discrepancy between the no humidity forecasts, which are in gray, and the ones that are generated with some form of humidity inclusion. Now, when we actually set this up for a comparison, and we used what's called a Friedman test followed by a Nemenyi test, which is in which we actually rank all the paired forecasts and see which one does the best for each week of forecast. What you can see here in the colors shows you the concentration of rankings, first being best, fourth being worst, for each of the forecasts in terms of their error for each of these metrics, predicting the peak intensity and predicting the timing of the forecast, of the peak, when it will be. And what you can see is that the no humidity forcing forecasts do the worst. There's a huge concentration around that fourth thing there. Does this actually work? Right around there, you can see it, and there. And actually the climatological forecast performs statistically the best. And actually inclusion of synoptic variability, that high variability that you get with changing weather patterns due in the daily humidity record, seems to actually destabilize the ability of the filter to optimize the model. So the forecast approach that we've been using seems to work best. In conclusion, basically influenza survival, transmission, and seasonality appear to be modulated by ambient absolute humidity conditions. We still don't know the mechanisms of why. There are a lot of hypotheses out there, and I'd be happy to discuss them with you. Accurate influenza forecasts can be generated using techniques that are borrowed from weather prediction. This is not really a weather-based forecast. This is a forecast that uses the methods of numerical weather prediction. However, humidity forcing does improve influenza forecast in this system, so inclusion of it is warranted. Thank you.
We have time for one question.
Yes.
Have you guys looked at other possible meteorological factors such as dust or aerosol or others?
Well, we certainly looked at temperature and relative humidity. We have also tried running the model in a climatological sense, as I did to describe the overall cycle, using vitamin D modification and also school cycles, which are very important for mixing patterns. Looking at dust and pollutant interactions is not something that we have yet done, but it's certainly possible that it's modulating it. I mean, this really gets to the core of once you expel the virus from the body and it's aerosolized or on surfaces right here on the microphone, what have you, what's actually affecting its survival? Is it salt toxicity? Is it pH? Is it humidity? Is it osmotic gradients? On the molecular level, we really need to figure that out.
Thank you very much. Next, we have Adrienne Thompkins presenting Assessing the Impact of Model and Climate Uncertainty in Malaria Simulations for the Kenyan Highlands.
I brought the water up because I was actually ahead of the curve for the forecast. So last week I was, shall we say, 4 weeks ahead of your forecast for the infection cycle, so to speak. So if I start coughing, excuse me. Okay, so what I'm going to do is show you a very simple study looking at malaria in the highlands. And with this simple study, I want to indicate that possibly climate uncertainty is something perhaps we should be focusing on often even more than, should we say, parameter uncertainty in models. So I presume it's down here. So of course malaria has a whole host of drivers running from not just climate variability, mobility, infrastructure, access to healthcare and so on, which both impact hazard and vulnerability. But I'm just going to be focusing on this small part of the puzzle of basically climate variations driving, should we say, changes in malaria hazard. Okay, and if you have a model of that system, it's going to be subject to a whole array of uncertainties. So we saw very nicely in the last talk, talking about uncertainties of various aspects of that influenza system. So we will have, I show here, basically errors and uncertainties in the climate information. Now, of course, if you're using a seasonal forecast model, that would be modeling errors itself. But even if you're using observations, there'll be the measurement error, but probably that will be dominated drastically by spatial representative errors. If you have a catchment area, complex topography, you only have to move a couple of 100 meters up or down to change the temperature experienced by the vector quite substantially. So a couple of 100 meters, you're talking about more or less roughly a degree. We're going to have initial condition uncertainties because you don't know the entomological or the hydrology situation at the start of your, should we say, forecast or simulation. And you'll have model parameter uncertainty and also model structure uncertainty. So the last talk was talking about structure and, should we say, structures of take into account absolute humidity or relative humidity and so on. And of course, there'll be inaccuracies in your health data itself, okay? So Madeline and I just made a very, very simple experiment where we said, okay, if we use a calibration technique, I'm just going to talk very briefly, it's a very, very simple technique, basically like a particle filter, a genetic algorithm, to try and calibrate the system. We can calibrate the model, okay, the parameter settings, but we can also use that same approach to try and calibrate all of the aspects of this hierarchy. So we can calibrate the, for example, the driving climate within its prior uncertainty, the initial conditions, or indeed the model parameters. And we simply want to see if we do that one step at a time, which will lead us to have, should we say, the closest fit to the malaria incidence for this highland location. Okay. So the calibration, so the model we're using in this study is the ICDP malaria model. It's just a compartmental model, an SEIR model for the disease progression in the host, but it's got a vector parasite life cycle model bolted on the side. So it has a compartmental approach to represent the sporogonic cycle, the parasite development, and the egg development, the larvae development, And basically, temperature is then explicitly built into that, in terms of the gonotrophic cycle for the egg development, the parasite development. So these are all parameters where the default values are taken from lab studies in the literature to represent how temperature speeds up this cycle, but also how temperature, as it gets too high, impacts the mortality of the larvae and the adult vector. We also have a very simple representation of the hydrology where rainfall drives a simple puddle model. I'm from a climate background, so it's a little bit like a cloud cover parameterization on the ground to represent the growth of the puddles, and intense rainfall flushes larvae. Okay. And so it's just similar to a genetic off-the-shelf kind of approach for the calibration, where we simply have a large ensemble of these models. We typically use an ensemble size of 80 or 100 models. They all have should we say, a selection of parameter settings, and we essentially just breed the models, repeating the simulations over time, where we select the best models that pass on their parameters to the daughter, should we say, generation. Okay, and those parameter settings can undergo mutation. So we do exactly the same thing for the climate. Sometimes we fix the model parameters, and we basically do the same thing for the driving climate by adjusting the trend or a small offset in the temperature within its uncertainty. And we also have a cost function, which is fairly standard for the departure, a little bit like a 1D variational simulation. So if we move too far away from the default parameters within our prior uncertainty, we penalize the model. So on the right-hand side, the red dot indicates like the most skillful fit to the data, and on the left, the default model. And in the middle, you have the kind of most likely set of models that the calibration tends towards. Okay. So we initialize the simulations just with artificial, should we say, conditions, and we spin up with 5 years. And what we're trying to simulate is just this T estate cases data, slide positive cases from 1979 to 2005 for a tea plantation in the highlands of Kenya. Okay, we apply a small offset to the data. There seems to be, we need to understand why there's a jump up in the last 10 years, which we believe might be associated with irrigation. So we're off to Nairobi soon again to meet to some of the people there locally and in the project and find out more about that. So the standard model with the standard settings basically doesn't simulate any malaria at all. It's too cold. The mean temperature there at the local station data is 17.3 degrees. So you can see most of the months, the inner two quartiles are basically sitting below the threshold for falciparum. Okay. And so if we don't get any malaria at all. When we calibrate the model, this is the kind of, should we say, the typical kind of simulation that we have. We have a pointer here. And you can see that it's not brilliant. Okay, so you can see some periods where we get very good representation of the seasonality. That's too far away to point over there. Sorry, everybody on the left side. We get the outbreak in 1999, but you can see that there are some misses where basically outbreaks are not represented at all. The correlation ranges, depends on the experiment, but between 0.25 and 0.3. Okay, so it's statistically significant, but you can see, particularly, for example, the beginning, there's a bad mismatch. This might be to lack of rainfall sensitivity because we're not testing structural errors in the model. There could be a whole host of reasons for this. Okay. So indeed, if I were to show you this, the light line here is the same plantation data, okay? And the black line, for example, if we was to say this was a simulation, if we look at the right-hand side or here, you can see that the black line here also misses a lot of these, should we say, outbreaks. But in fact, this is just, this is from Shanks et al., and this is actually a plantation right next door. So the point here is just that you can't explain climate variability, right, as I said at the first slide, to explain all the outbreaks. These two plantations are right adjacent to next to each other. You expect the rainfall to be the same. It may be that one is slightly higher than the other, so the temperature may be slightly different in those two plantations. So you can't explain everything by climate, of course. If we look at those simulations, we just did a quick wavelet analysis. And the model indeed is far too regular in the outbreaks. On the right-hand side, we see a strong peak in basically the annual cycle, and there's a much stronger peak associated with the ENSO variability every 4 years in the model runs compared to the data, whereas the data, should we say, has a much wider, should we say, peak in the power spectrum across 6 to 12 months. Okay. And I think this is especially bad because 2 of the January-February, should we say, outbreaks were missed in the model simulation. So if we try to separate now this into that first run was just calibrating both the model and the climate. If we actually split this up into model calibration and climate calibration, on the top here, all of the parameters, if I have 2 or 3 lab studies, I try and estimate the uncertainty for the prior from those. Otherwise, I have just set the model uncertainty for the sporogonic and gonotrophic cycles to 20%. And you can see that within that level of uncertainty, we do a very poor job of just calibrating the model. If we just calibrate the climate, then basically all of that fit to the data is basically coming just from the calibration, mostly from the calibration to the climate. So this primarily is just from offsetting the temperature by just over a degree. Just warming, a constant offset to the temperature, is enough to go from no mirror simulations to actually be able to get some of this variability in some of these outbreaks. So in this location, the climate uncertainty is far more important than the model calibration. But of course, the caveat for that is it's very difficult to specify these priors, the uncertainty in these parameters from the literature, because often you only have 1 or 2 lab studies actually examine these parameters. Okay. So I also want to emphasize that this result is probably specific to the location where we are, and I can illustrate this. If we just look at, for example, the sporogonic cycle, so basically the degree day length, the amount of time it takes for the parasite development to actually occur within the adult, if we actually play with the degree day length on the top here, so we just say, okay, plus or minus 20%, that's the black bars here, whereas plus or minus 1 degree uncertainty, of course, if you're very close to 18 degrees, then adding 1 degree makes a huge difference in the sporogonic cycle length, whereas you go to warmer temperatures, the model uncertainty and the degree day length actually dominates. So it's a result that we expect, and at warmer temperatures, then you would suspect that model uncertainty would become relatively much more important, not just for this, but also due to the gonotrophic cycle. Okay. Quick mention, how am I doing for time? I don't know where the clock is. 3:44, okay, right in front of me, there we go. Just a very quick slide on the initial conditions. When we tried to calibrate for the initial conditions, we just basically didn't get any convergence at all. And the reason why is that if you actually look here, I've run basically simulations that start from an initial parasite ratio ranging all the way from 5% up to 95%. And you can see that all of these simulations basically converge after about 2 years. So effectively, everything's controlled from the boundary conditions. The set of equations are essentially asymptotically stable. Everything converges over time. Now, if you think about this, in fact, this, again, I said I'm from a climate background. This is exactly the kind of behavior you see from a regional climate model, where everything is constrained by the boundary conditions. We always think of like NWP models of after your 5-day timescale, essentially you have exponential growth of initial condition errors. But for regional climate models, that's not the case, because everything's strongly constrained by your boundary conditions. And the same thing's happening here. Your boundary conditions are the climate. So your climate envelope constrains what's going on, and the should we say the prevalence modeled basically converges after about 2 years. So of course, if you want to do a seasonal forecast, the initial conditions are critically important. So the hydrological situation and the entomological situation, but for these long-term runs, that's not the case. Okay. So in this simple preliminary study, I just wanted to show that this technique where we're using this, should we say, this simple particle filter approach to calibrate each layer, so the climate uncertainty and the initial condition uncertainty and the model parameter uncertainty. And we found that the climate uncertainty was by far the most important for simulating malaria in the highlands. Okay. But of course, there were quite a few caveats here. The definition of the priors are certainly very ad hoc. We have perhaps a poor sensitivity of rainfall in this model. There's no migration in this model, and so on. There's lots of processes that are neglected. Okay. But I think the approach offers a very simple integrated framework to try and analyze these uncertainties. People have used calibration to set model parameters, but I think it's quite nice to try and think about all of the layers of the model system in the same framework to try and compare how the uncertainty compares in all of those. So anyway, thank you very much.
We have time.
At the end without coughing.
We have time for a couple questions. Anyone? Yeah.
Very interesting presentation. Very striking that the best thing you could do was just to add 1 degree.
Yeah, it was about 1.2. Yeah.
Model.
Yes.
Suggests, first of all, microclimates are the driver of these diseases. And are you looking at ways to do something more sophisticated than adding 1 degree to be able to make it maybe more generalizable to different areas? Or?
Right. So, I mean, in that case there, you're exactly right. So the idea of that, so we had a prior uncertainty of about 1.5. And that's basically saying exactly that you can have topographical changes. So in a kilometre, you expect a 6.5 degree variation. So if over your plantation there's a change of 100 or 200 metres, then you would expect for a 200 metre change on the order of 1.3 degrees or so. But also land cover can also change. You've also got differences of, should we say, in hut and outside hut temperature and all these kind of things. So the idea is then to allow, should we say, a little bit of wiggle room to represent those uncertainties. Now, the calibration, of course, would choose a different offset in each location. I mean, ideally, for example, if you have a coarse scale grid, so when we use this model for seasonal forecasting in Uganda, then we do at least try and use a topographical downscaling to go to a much finer grid in terms of the temperatures. But then I still think it's very helpful then to actually apply this on top of the calibration down to your local, should we say, district health centre's catchment, if you know that. Sorry, a little bit of a waffly answer. Do I have time for one more?
No, we'll have them at the end.
Okay, no problem. Thanks.
Thank you. Next we have Jeff Shaman again, presenting in lieu of Teresa Yamana, a presentation titled Environmentally Driven Ensemble Forecast of Dengue Fever.
So the talk actually is going to be a little bit different because where Teresa had gotten with some of that work was not quite as realized. So I'm actually going to talk about a different postdoc, Nick DeFelice's work, looking at another vector-borne system, West Nile virus, in which we're doing something similar to what I talked about with flu and developing forecasts of it, and we're actually looking to see whether or not we can do something mechanistic with temperature forcing to improve the forecasts. Want to get this out of the way. We have a lot of people we cooperated with on this. A number of mosquito abatement and health districts provided data and time and expertise talking to us about this, as well as some other co-workers within it, and obviously a bunch of funders there from DoD and NIH. So West Nile virus, unlike malaria, is maintained in a zoonotic transmission cycle in which the principal hosts are avians, birds, and Culex mosquitoes are the primary vector in the United States. Basically what happens is an infectious mosquito bites a bird, passes the virus onto the to the bird during its consumption of the blood meal. It just effectively spits into it. The bird can then get infected, run a high viremia, and then pass it back to another mosquito upon feeding a few days later. This cycle can perpetuate, and the cycle that you see there. Humans and horses and a few other animals are referred to as dead end or spillover hosts. We can actually become infected if bitten by an infectious mosquito. We can get quite sick and it can even kill us. However, we tend not to run a high enough viremia in our peripheral blood system to pass the virus back to mosquitoes, so we're not part of the transmission cycle. So we've developed a forecast model or system that's similar to one I described for influenza. It's going to be made up of 3 components: a model describing and capable of free simulation of West Nile virus transmission, data streams that we can feed into it in real time in the hope of real time in order to actually optimize the model, and an algorithm, a data assimilation method. Again, we're going to be using the ensemble adjustment Kalman filter because it's actually quite efficient for optimizing the model on the fly. The data that we used for this, for the initial component of this, is we got human case data on a weekly basis, I should say, as well as mosquito infection rate data from mosquito abatement districts in 2 counties, Cook County, Illinois, which is all around Chicago, basically Chicago's in that, and Suffolk County, New York, which is on Long Island. So for the mosquitoes, they trap them. They have multiple traps throughout the counties. They then sort them by species. They then send them off in pools to be ground up and tested for West Nile virus. You get for each pool whether it's positive or negative for the virus. And we then have to use a maximum likelihood estimation technique to estimate the number of infectious mosquitoes based on the number of pools that have come back positive in a given week, et cetera. So that generates a time series of data for infection rate in the mosquito, and we have the human case infection rates from hospitals in the area, because West Nile virus is a notifiable, reportable illness. The model that we built, which is the baseline model, this lacks temperature in it, is fairly parsimonious. It's 12-dimensional. It's got 5 state variables: susceptible birds, infected birds, susceptible mosquitoes, infected mosquitoes, and then infected humans that are spilled over too. Has a bunch of parameters in it that guide the death of the mosquitoes, the bird recovery rate, and a complex logistic function for the contact rate between birds and mosquitoes that actually emulates the fact that these mosquitoes over time transition over the course of a season, I should say. From early season, they tend to feed principally on avian hosts, but they move towards mammals as you get into the fall, which is of course detrimental if you're a human and don't want to get West Nile virus, because they start biting humans preferentially. And then the third ingredient is, again, an assimilation algorithm, and I won't go into too much detail of it here, but I will tell you a little bit about how we test the system, which is something that we do for all our infectious disease forecasts. We want to know how well the filter is actually optimizing the model, and the way we do it is something called a synthetic test. Most of the state variables and parameters within the model, and here it's only a 12-dimension system, we are not observed. We don't observe the number of infected birds, we don't observe the number of susceptible birds or mosquitoes, we don't observe the parameters guiding the force of transmission and the mortality rate of mosquitoes. These are things that we want the filter to be able to estimate in the absence of observations through the data assimilation procedure. What we can do is we can run the model in free simulation and generate a synthetic outbreak, which is the blue line there. We then take the 2 variables that we would observe, in this case human cases and infectious mosquitoes, and we take those time series, which are shown here in the cyan color right here and here, and we add noise to them. There's a lot of noise associated with mosquito observations. It's really kind of like a negative binomial process. So you can see there's a lot of variability on what we would expect to observe from maybe 50 traps being run in a given week in a given county. And then there's not quite as much, but still some noise in the human case data. We then feed that noise data as our observations back into the model filter system. And we see, can it actually estimate the unobserved state variables and parameters? How well is it actually optimizing this nonlinear system over the course of training? So this shows you one example of it here. And what you can see, again, the blues are the truth. These are for the 2 observed variables we had, infected mosquitoes and human West Nile cases. The little red things, these are little tiny compressed box and whisker plots for multiple posterior simulations of the system, and you can see we're doing a pretty good job of estimating the truth. Now remember, we fed in those noisy observations, synthetic observations. Here are the other state variables, and you can see again here, we're doing very well. And we have no information on it. We're only giving information to the system about 2 observed quantities. The ones that you saw right here in these blue dots is all that was given to it. But we're getting a good estimation of the susceptible mosquitoes, susceptible birds, and infected birds. Furthermore, we're able to estimate some really key epidemiological parameters within the system. This is the contact rate, which is that complex logistic function. This is what it looks like over time as it was imposed, and here is the estimate of it, again, only born from giving it noised up information from the top row. Here's the human spillover rate, the rate at which transmission is going towards humans. You can see it doesn't do as well, but it's moving towards the truth there. So we're actually getting some decent evidence of optimization of this system, which suggests that we might be able to use it in forecast, because when we use it with real observations, we can't do the synthetic test. We don't know what the true targets are, but we have a sense from this that it's converging to the truth, hopefully. Hopefully, I do say. So for the forecast, what we do is we use our weekly observations. If let's say we're in August, we're going to take all the observations of infectious mosquitoes and human West Nile cases up to our point in real time in August. We're going to assimilate each weekly observation into the model and adjust the model over time. We run the model as an ensemble of simulations. Once we get to the last observation and we update the system for the last time possible, we then run it in free simulation into the future. The whole simulation provides a prediction interval on our forecast, and it's run to the end of the season, and that is the forecast that we indeed generate. So this is what it looks like for retrospective forecasts 1 year. This is for 2010 in Suffolk County in New York. The X's right here are the observations that we have in hand. The little stars in orange are the observations that we don't yet know about in the system. The posterior is the thick blue line that matches the length of the observations, and the forecasts are these dashed lines here. Now we've run 5 of them, which is why you're seeing 5 of those lines there. The grays are credible or prediction intervals around that. This is the interquartile range, and the lighter gray is 10 to 90th percentile. In addition, we're going to look at the accuracy of it, and we're going to see whether or not we can predict the total number of human cases within plus or minus 25%, and that's these lines right here that it's showing you around the final observation. So how well do we get that? And then we're going to look to see if we can predict the peak timing within plus or minus a week, and the maximum number of infected mosquitoes within plus or minus 25%. And that's that green box right there. So I'm going to step you forward through weekly forecasts as they're generated in retrospect. And what you're going to see over time is that it's going to have a wide credible or prediction interval at first, but as more information is brought in, it starts to narrow. And we get to this point right here, and you're well ahead of the end of the season in terms of when you see human cases cropping up in the hospital. And what you're actually seeing is it's making a good prediction at this point. It's already well constrained by what's going on. You can step further along, and it stays to those points there, and we get very good predictions of human cases in the county, which is really our principal goal right here. All right, when we look at this over all the seasons we did it for these 2 counties, we can make these plots right here, which are sort of a distillation of the overall accuracy of the forecast. We're looking at what fraction of predictions were accurate from 0 to 100% right here, 0 to 1. And then it's as a function of the predicted lead week versus where the mosquitoes peak, where that peak infection of mosquito occurs. So this is when the peak of mosquitoes occur, and it's actually after this for about the next 6 to 8 weeks that human West Nile virus cases really start showing up. It starts a little earlier, maybe -2, but 75% of the cases, human cases, occur right here. And what we can see in particular is that we have reasonable predictive accuracy. 50% of forecasts and going higher can predict this peak timing a bit beforehand. But more importantly, if you look at the blue line for the human cases, you can see we're not getting very good predictability here. Only about 20% of the forecasts are accurate. But then once you get to the peak right here, right around it, it really starts to ramp up. But again, I want to remind you that most of the human cases haven't been observed then. So this is actually up to 9 weeks in advance of when you see the final human case. We're getting decent predictions of the level of the amount of human cases that are going to wind up in hospital within the county. So we have this baseline system, and we wanted to actually see, could we impose some sort of environmental forcing on it and improve the system? Temperature is the way we went with. We looked at temperature, which influences a number of things about this system. It affects mosquito development rates. It shortens the gonotrophic period, which is the time interval between host seeking. It decreases the extrinsic incubation period of the virus, which means the virus is going to develop more quickly as it gets warmer. But warmer temperatures, if they're excessive, will actually decrease vector survival as well. So there are a lot of relationships in here, and they're all actually nonlinear. A lot of them have been borne out from laboratory studies. We actually focused on the extrinsic incubation period of the virus, and we posed it on this contact rate. So rather than having just that logistic function I showed you before for the transition between feeding on birds to feeding on mammals right here, this is the contact again between birds and mosquitoes, we have a temperature modulation of that. Whereas it gets warmer, they're going to be more likely to be feeding, period. As a consequence of this, you're getting something that looks more like this for a typical temperature cycle. And we're going to compare the 2 models, temperature forced and not temperature forced, but at this point we were able to expand the number of groups we were working in to include 12 counties from around the United States that were able to provide the same data. When we did this, we get something that looks like this for each of those 4 metrics I showed before. Let's focus just on the top 2, human cases and peak timing here. What you can see for the red line, which shows the accuracy, the fraction of forecast accurate as a function of lead week, the temperature force models in red are doing better. For the entire time period, regardless of lead, for peak timing of mosquitoes, and for much of the time for human cases, particularly in this period right here when they're just starting to ramp up. You really don't have very many cases here. This is about 85% of the cases here. So we're getting a better forecast with the temperature forcing, which suggests that the biological use of it and the biological plausibility actually is improving forecasts by using those conditions. Now, I should add again, we have to use climatological temperature. We don't know what it looks like 9 weeks in the future with temperature, and we've done some experiments on this as well as we did for the influenza, and a climatology, daily climatology actually works quite well. So I don't have much time, but just say we actually started doing this in real time. We forecast 4 counties in real time this past season. One of them, Cook County, allowed us to publish it online. The other 3 did not want us to. We just provided the forecast to the mosquito abatement districts. There are a lot of issues associated with this. There are huge lags between when the mosquitoes are collected and when you actually get that West Nile virus positivity rate, which is problematic. Human case reporting is also not very good. Even though this is a required notifiable disease on a national level, there are very large delays in when the cases are reported. It can be 4, 6, 8 weeks after somebody actually has been diagnosed with West Nile virus that it finally winds up in the system. That's very problematic when you're using that information to assimilate. So if we're going to do real-time forecast of a system like this, we need expedited production of this data that's a little more accurate. So anyhow, that's what the real-time forecast for Chicago looked like. And there's the summary slide, but I got 5 seconds, so I'll stop there. Thank you.
We have time for one quick question.
You're on again.
Yeah.
In your experience, what has practitioners done as most important getting that forecast? The timing correct or the magnitude correct?
You mean of the data, of the observed?
Sorry, of the forecast.
Oh, what of the forecast is appropriate for getting that? You know, we haven't actually broken it down that way. The system is linked. I think it would be maybe a little challenging to do so. I mean, the whole system is a nonlinear model, so it has its own complexities. We could break it down statistically and look at, you know, do we do better when we're doing a better peak timing versus peak magnitude forecast, but we haven't done so. That would be something interesting to look at, though. Thank you. Thanks.
Next we have Amir, Siraj. Oh, I'm sorry. We have Jamie Caldwell presenting predictive modeling of mosquito abundance and dengue transmission in Kenya.
Thank you. So today I'm going to tell you about some research that my collaborators and I are doing investigating ecological drivers of mosquito-borne diseases that are transmitted by the Aedes aegypti vector, which is pictured here. And the focus of this research is dengue, a disease that affects over 390 million people annually. And it has a variety of symptoms from a headache or general malaise to joint pain, severe fever, and even in some cases, death. And without a commercially available vaccine, our best source of disease prevention is mosquito control. And intuitively, this makes sense because the more mosquitoes you have, the more mosquito-borne diseases you have. But the relationship is actually a little bit more nuanced than that. And thus, a major focus of our research has been understanding how the distribution and abundance of mosquitoes actually relates to human case data. And if we think about this mechanistically, we know that a series of climate factors, such as temperature, rainfall, and humidity, affect many traits of the mosquitoes themselves and the viruses they transmit. And so our overarching goal is to understand the climate drivers of vector abundance and transmission, and then use that information to make predictive models to understand the transmission dynamics in space and time in both the mosquitoes and humans. So the framework that we're using for this research is another compartmental model, an SCI-SCAR model. And this is a visual representation of those differential equations, where we can model the movement of disease through the human and mosquito populations. So in the top red circles, we have the human population, which is split into susceptible, exposed, infectious, and removed classes. And in the green circles on the bottom, we have our mosquito population, which is separated into susceptible, exposed, and infectious classes. And the solid arrows show the movement of individuals among classes, and the dashed arrows show the directionality of transmission. So for example, for individuals to move from the susceptible to exposed class, they need to get bitten by an infectious mosquito. Now this isn't a new framework. Many labs have been using it and doing really good work all around the world. But what is new and is exciting about coming out of this research is that we're able to incorporate temperature dependence into that model. Because we know from many laboratory studies that temperature affects a suite of processes associated with both the mosquitoes and how they transmit disease. So if you look at the graph here, on the y-axis we have mosquito reproduction, and on the x-axis we have temperature. And the black dots are from a meta-analysis of multiple lab studies. And what's important to note here is that the relationship is unimodal. So optimal temperature for mosquito reproduction is somewhere around 29 degrees. And if you're either above or below that temperature, reproduction is lower. And for all the temperature-dependent traits that we know about from laboratory studies, we see similar unimodal relationships. So that includes traits like the mosquito development rate, so the time it takes for the mosquito to develop from egg to adult, mosquito density, the rate of biting of mosquitoes on humans, the virus development rate, so the time it takes between the mosquito getting infected and becoming infectious, and even the lifespan of the mosquito. So using this new temperature-dependent model, we can predict, given a location's mean temperature and temperature variation throughout the year, what the epidemic suitability is. And this is a heat map of locations where the Aedes aegypti vector exists. On the x-axis, we have mean temperature, and the y-axis, the temperature range. The dots are showing just 20 major cities worldwide and where they fall on that heat map. The colors represent epidemic suitability. And what you'll see is a band of deep red where epidemic suitability is highest. And what you'll notice is when there's lower range in temperature throughout the year, it's more optimal for epidemics in warmer temperatures. Whereas if you get greater variability, it's still really suitable for epidemics, even when you have lower mean temperatures. So what we wanted to find out is, does this model actually reflect what we're seeing on the ground? So this is a map of the 4 sites in Kenya that we use both mosquito trap data and human case data from. And this data has been collected since 2014 and will be ongoing through 2018. So I'm going to show you some of the data we have using one site in Western Kenya, Chilambo, as an example, but I'll show you the results for all the sites. So first we have temperature loggers at all the sites. This is a profile of daily temperature, rainfall, and humidity from loggers. What you can notice here is that there's some seasonality, but for temperature especially, there's low variability, whereas you have higher variability for rainfall and humidity. For the mosquitoes, we have 20 sentinel houses at each of our sites, and at those houses we have mosquito traps collecting data on the 4 mosquito life stages pictured in the cartoon in the lower left. So if we look at an example from one house, we'd have data that looks like this. On the y-axis, we have frequency of the different mosquito life stages, and on the x-axis, we have time. And the bars here represent a single monthly sample because we set out the mosquito traps monthly. And here again, you'll see some seasonality, but everyone hear me? But the major seasonality can really be seen in the eggs and adults here. And finally, we have human case data. So we have about 500 kids enrolled in the study from each site. They enroll when they come into the hospital with a fever and are suspected to have dengue. And blood samples are taken, which can be seen in the picture from one of the PIs from the project. So blood samples are taken when they initially come into the hospital, and then 1 to 3 months later. And each of those blood samples are tested in 3 ways for dengue positivity. 2 of those tests test for virus in the blood, and 1 for antibody to show past exposure. And so the result is this time series of dengue cases. So now that I've shown you the data, I'll tell you a little bit about how we analyze this. So we take that temperature profile on a daily basis and run our mechanistic temperature-dependent model, which gives us predictions of both the human and mosquito population over time. And what we were particularly interested in this study is the infected number of people over time and the total number of mosquitoes, because we wanted to match this model with our observational mosquito trap data and human case data. So the way we did this for the case data, because we're only getting a subsample of the population, we can't compare these directly. So we used a logistic model to determine what's the probability of any given child being infected, given the predictive proportion of the population from the model. And we took a randomly selected 80% of the data set to develop the model and use the withheld 20% to test the fit. And we bootstrapped this process 1,000 times to make it more robust. For the mosquito trap data, we used a linear regression to connect it with the predicted infectious or predicted mosquito population, and similarly, we used training and testing data sets and a bootstrapping process. Now, the total number of mosquitoes predicted from the model is meant for adult mosquitoes, but we ran this analysis with all 4 life stages. And the reason we did this is because the traps have different efficiencies, and so we thought it was possible that one of the other life stages might have been more representative of the mosquito population. And finally, to determine fit of these models, we evaluated the predictive capacity compared to a generic null model. So is it predicting mosquito abundance and dengue cases better than random, so 50% of the time. And then we used an informed model. So is this temperature-driven mechanistic model better than a temperature-independent model? Do we need this added complexity? So on to the results. This is a frequency distribution of the area under the curve for the dengue case data. And for this measure, 1 would be a perfect fit. And so I want you to first focus on the orange distribution. That's our temperature-dependent model. What you can see is that we can accurately predict dengue incidence about 60% of the time. Now, this is better than that generic null model of 50%, but only slightly better than our informed null model, which doesn't account for temperature variation. On closer examination, we can also look at the strengths and weaknesses of this model. And so for our purposes, we're mostly interested in that true positive rate because we want to be able to predict dengue cases when they actually exist. And so what we see here is that the temperature-dependent model does in fact slightly increase our true positive rate compared to our informed null model or a generic null model. So moving on to the mosquito results, we have another frequency distribution, but we're using a slightly different metric of fit, which is root mean square error. So for this measure, a better fit is represented by a value of 0. So what we can see here is that the best predictor is actually pupae, followed by larvae, whereas eggs in adults don't actually have a great fit. What you also might notice is we don't have a second distribution here of the temperature-independent model, and that's because the distributions actually overlap perfectly. And so this was really surprising to us that the temperature didn't improve our model fit for the mosquitoes. But what we think is happening here is that Kenya actually has fairly stable temperatures throughout the year. So if we go back to our graph of this epidemic suitability, Kenya falls in this gray box, in which case we shouldn't really expect a lot of the variation to be accounted for by temperature only. However, we're also going to be testing this model and validating it with data from several sites in Ecuador. And here there's lower mean temperature and much more variability. So it will be really interesting to compare the results from these 2 data sets. So overall, we found that this mechanistic model was able to predict mosquitoes and human case data with some accuracy. And what's exciting about this is it's not a statistical model, it is a process-based model. So this is telling us that we're capturing some really important mechanisms that are associated with disease transmission. However, for here, in this case, the temperature-dependent and independent models didn't differ that drastically. And again, we expect that because of the temperature range in the Kenya data set. What was really interesting to us as a group is the generic obstacles to model validation that was brought up a little bit in the last talk. Even though we had all the components to really test whether this was a good model, it was difficult to connect the mosquito data, the human case data, the model, and then determine the right way to really rigorously determine whether we were doing a good job at predicting these data. And finally, I just want to say that although temperature drives a lot of these processes, we know it's not the only important factor, and so the next step is going to be incorporating rainfall, and after that, humidity. Thank you.
Do we have one or two questions? Yep.
So we haven't looked at it yet, but I will just say my hypotheses about this. I think that temperature is really important, but it's important given that there is enough water in the system. And so I expect what we'll find is, again, a nonlinear relationship, but that will be really important for determining when the temperature matters. And so we expect both that low and higher rainfall conditions will find more because people store water when there's drought conditions, so we expect a relationship like that.
Any other questions? I think Gabe had a question.
So, this is a general question, I think, for a lot of the speakers. When you look at temperature, I've seen a lot of daily mean temperature, but and you look at the annual cycle and seasonal variations about it, but the diurnal cycle in many of these places is quite large. Does it matter at all? Is the daily mean the relevant temperature, or is daytime or nighttime temperature more relevant?
Yes, that's a great question. And actually, the model that this is based on and that meta-analysis I presented was done from my supervisor, and they looked at daily temperature and other measures. And actually, the most relevant one was average daily temperature. But yeah, I was actually surprised with that result too.
Right.
Thank you.
We'll go on. Next, we have Amir Siraj presenting temperature modulates dengue virus epidemic growth rates through its effects on reproduction numbers and generation intervals.
Thank you for the opportunity, and thanks everyone for being here. My talk is going to be on theoretical data rather than theoretical aspect of dynamics rather than empirical. I'm going to look at the temperature effect on the epidemic growth rate rather than intensity of the transmission. So several studies have focused on the effect of temperature on the transmission potential rather than the speed of the epidemic growth. But we think speed of the epidemic growth is one important aspect that has been overlooked for quite some time. So we wanted to look at that aspect of transmission. These are some studies that have focused on the transmission potential effect of temperature for many reasons. Why do we focus on arboviral diseases? Because arboviral diseases have been growingly threatening global health, and they come in episodic kind of transmission dynamic. So the speed of their transmission potential is really an important aspect of the dynamic that needs to be estimated for effective public health intervention. And the other thing is early estimate of the epidemic growth rate really gives an important estimate of the number of cases that could be estimated within a week, within a month, etc. The rationale being very tiny change in the epidemic growth really ends up in getting quite diverse epidemic outcomes. For instance, in these two curves, the top one being linear, the bottom one log scale, a difference between 0.4% and 0.5% epidemic growth would give us order of magnitude different number of cases within 180 days. So it is important to be very specific in terms of quantifying the epidemic growth rate. And also, estimating the epidemic growth rate is achievable because, one, we have we can estimate temperature-driven parameters like all the other talks have discussed. Also, we can use a very basic relationship between three quantities to basically arrive at the epidemic growth rate. One, basic reproduction number, generation interval, and epidemic growth rate. I will come to each of them in my next slide. Let's look at basic reproduction number. It's the average number of secondary infections in human originating from a single primary human infection introduced into a fully susceptible population. So basically the transmission potential, which usually is the most used parameter and the most used in terms of quantifying through temperature effect. We use three parameters to quantify our real basic reproduction number. One, incubation period, biting rate, and mortality rate. We use the Rose-McDonald setup for R0 to estimate temperature-driven R0. The second component of our equation is the generation interval. It is the time between two consecutive infections. So basically, when a person is infected, the incubation period kicks in and then the infectious period follows. That's when the mosquito can pick up blood from the human and possibly transmit it to the second human host. This is if, because it has to stay alive for the period to complete and for it to pass it to someone else. So because of that iffiness, in all of them, actually in the incubation period, intrinsic incubation period, in the infectious period, and in the extrinsic incubation period, and in the period between the mosquito being infective and transmitting it to the second human, all of them are probabilistic. So we approached it probabilistically and estimated the whole generation interval based off of probabilistic approach, which is shown here. So the first one being incubation period, we used literature, which at about average at about 6 days for dengue fever. We used human to mosquito transmission period. That's the period between the mosquito being the human host being infectious and the mosquito getting infected. We estimated that also from literature, which arrives at about 3 days average. And then we estimated the extrinsic incubation period also from literature. This is temperature dependent. So we have to estimate distribution for each temperature value, basically, going from lower than 20 to all the way 40 degrees Celsius. Similarly, we estimated the mosquito to human transmission period. Probabilistically, we estimated this based off of the probability that the mosquito will survive to transmit it to another person. And then we put together all the four sequential temporal aspects of the dynamic to get a one generation interval quantifying the time between two consecutive infections. So what do that give us? So put together the reproductive number, the generation interval. Intuitively from population dynamics, we know for a given generation interval, increasing, for instance, R0 would increase the epidemic growth rate. Also, we know that for a given generation interval, for a given R0, shorter generation interval will give us a higher epidemic growth rate. But we wanted to quantify specifically. So we used this approach from population dynamics, which basically relates the three quantities. R0, little r, which is shown there. Little r is here, and GA is the generation interval distribution. So if we integrate over the total, the entire generation interval, we can quantify R naught with this equation. Because we know R naught from temperature-driven estimate, and because we know the generation interval distribution from temperature-driven estimate, we can solve for little r. We may not be able to solve it analytically, but we can solve it numerically. So that's what we did. So to go to results, this is the generation interval distribution. Every vertical line is distribution, generation interval probability distribution. As you can see, at 25, it's wider. The more it goes to wider, the more it gets wider, the higher the probability. So here it is wider, here is narrower, and here is higher. The solid line is the mean, the broken line is the median. So it goes from 25, for instance, to 35. It goes from 30 days to half that length of the generation interval. And it's nonlinear. It goes narrower here because of the effect of mortality at higher temperature. Results for the reproductive number also nonlinear. It goes to a certain peak temperature slowly, but collapses fast due to the effect of mortality as well at higher temperature. It peaks at 32 degrees Celsius. It doesn't change for any mosquito abundance value. We looked at a range of mosquito abundance value, and so the peak value doesn't change for any amount of mosquito abundance, but the value itself, of course, changes for a bit of the mosquito abundance. How about little r, our target? So interestingly, little r also goes non-linearly and peaks at about 33 degrees Celsius. And for any amount, for any value of mosquito abundance, it doesn't really change much. So it was very interesting to see that, which means basically we can use that peak temperature to differentiate between different regions of growth and collapse of epidemic growth rate. And that's what we did. I will come to that in my next slide. Before that, the peak temperature, we looked at the peak temperature, we zoomed in at the peak temperature, and we Monte Carlo simulated 1,000 samples of the parameters that we used to estimate R naught. And we see that the peak temperature actually is very robust within a very narrow temperature band. We looked also at the rate, the role of the two components of our little r calculation, the generation interval and the reproductive number. In red, it is at each temperature the ratio of contribution between the two factors. So as you can see, the red is generation reproductive number, and blue is the generation interval. Generation interval is very significant contributor in some very key temperature values like 32 degree, 32.5 and here, and 22.5 there. We also looked at off-the-shelf generation interval distributions, like exponential distribution, for instance. It showed that it overestimates epidemic growth rate. We also looked at hybrid, basically informing a fixed length generation interval with our temperature-dependent estimates that we used in our formulation. And that's when we could really come closer to our estimates. And finally, we used the fixed temperature threshold for epidemic growth rate, regardless of the mosquito abundance. We used that to basically look at areas of Aedes aegypti suitability and estimate how far, given the climate change scenarios, this one, for instance, RCP 4.5, how far will epidemic growth rate be affected? So, for instance, in the red region, it means because the temperature is now below the threshold value, and it will stay below the threshold value, epidemic growth rate is going to be worsened by climate change. And in here, in the yellow, the temperature is going to be shifting towards the area where more temperature means less epidemic growth rate. And the green one is the already past the threshold temperature value. So it won't change much. I mean, it would be in the area where more temperature means less epidemic growth rate. And so we looked at these regions and estimated their population in different scenarios. As you can see, most of the population falls in the first category, which is more epidemic growth rate will be the effect of climate change by 2050 for all three climate change scenarios. So the caveats, it depends on the current understanding of the relationship between temperature and these parameters, and specific values of the epidemic growth depends on specific value of mosquito abundance, which we don't have right now. In some settings, the effect of temperature may also be overweighed by effects of non-climate factors. Conclusion, fully specified generation interval was successfully quantified the epidemic growth rate. A robust temperature threshold of epidemic growth rate helped generalize the effect of across regions of varying mosquito abundance, and temperature change under future climate scenario is potentially gonna increase epidemic growth rates for many areas. With that, I acknowledge my colleagues, my co-authors, and funding sources. This work has been published recently in PLOS entities, and the code data we used to generate this functional relationship between temperature and generation interval, also epidemic growth rate and R0 are available at this GitHub site. With that, I take questions.
We don't have time for questions right now, but we'll take more questions at the end.
All right.
Okay, next we have Ludovica Beltrame presenting how do changes in hydroclimate conditions alter the risk of infection with fasciolosis.
Hi. My talk today is on a study that looks at how we can assess the impact of climate change on disease risk through integrated mechanistic modeling using the example of fasciolosis. And this is a joint collaboration between civil engineering, biological sciences and medicine at the University of Bristol in the UK. Recent reviews identify three main challenges in assessing climate change impacts on infectious disease. First, to disentangle the effect of multiple drivers on disease transmission. This is especially true for diseases that may include weather and climatic variables as potential drivers, but also environmental characteristics or landscape characteristics, which is true for diseases involving an intermediate host or vector as part of their life cycle. Second, to describe the mechanism between climate factors and disease transmission explicitly in order to be able to more reliably assess the impact of conditions beyond the previously observed variability on disease risk. And finally, to identify the relevant space-time scales for the system in order to capture relevant impacts and to provide decision support. So we want to look at these challenges using the example of fasciolosis. Fasciolosis is a widespread zoonosis, meaning an animal infection that can be transmitted to humans. It is caused by this 3 centimeter long parasite which infects the liver, hence the name liver fluke. Clinical disease in animals include, symptoms include anemia and can lead to sudden death as well, whereas subclinical infections can lead to lowered productivity and have been estimated to cost the livestock industry around $3 billion per year annually. And the disease is urgent to address for a number of reasons. First, there is no vaccine against it, and there is increasing resistance to the available antiparasitic drugs that has been reported worldwide, and it is emerging in humans with controlling humans relying on the same vet drug to which resistance is increasing. And risk of infection with liver fluke is strongly controlled by weather and environmental conditions, as the parasite has this complex indirect life cycle involving 3 living stages, which are these eggs, Myriacidia and Metacercaria, as well as an intermediate host, which is this mud snail, which live and develop in the environment until Metacercaria get ingested by the animal, infecting it. And temperature has a direct impact on the development and mortality rates of these stages. So, for example, eggs do not develop below a temperature of around 10 degrees Celsius, and below that temperature, also snails are usually inactive, whereas rainfall, the impact of rainfall on disease transmission is mediated by the environment through soil moisture. So, for example, hatching of eggs into Myriacidia only occurs with wet soil conditions, and snails can only be found in muddy areas with poor drainage. And in the UK, an increase in prevalence from 48% to 72% in dairy herds has been reported in the last decade and attributed to wetter and warmer conditions, as well as it has been seen an expansion of the disease into previously unaffected areas, such as Scotland and East Anglia, and a shift in the seasonality of the disease has been observed, making disease control challenging and raising concerns about the effects of climate change in the future. And existing fluke risk forecasting models are based on empirical relationships between historic climate and incidence data. Therefore, they are unsuitable to assess the impact of conditions beyond previously observed variability. And the Ollenshaw index is the most widely used. It was developed in the 1950s, but is still currently used to inform farmers about potentially high-risk season or years in the UK and in Ireland, for example. And basically, the model calculates monthly risk based on monthly rainfall and temperature. And despite being empirical, the model has been used to assess the impact of climate change on disease risk in the UK up to the 2070s at 25 square kilometer resolution based on the resolution of the available climate projections. And this approach is common also beyond fasciolosis. This is the result of a recent review that basically says that despite many diseases involving many drivers, especially those involving, as I said, vector or a host as part of their life cycle, most studies usually focus on temperature and rainfall when assessing climate change impacts on disease risk. And moreover, also as for fasciolosis, there are usually empirical associations between mean climate characteristics and disease averages. So instead of relying on correlation, we developed and tested a new mechanistic hydroepidemiological model for liver fluke, which we called HEALTH. The model comprises a hydrological part that is a catchment scale rainfall runoff model, which we use to simulate soil moisture, and we couple that with a model of the parasite, which is based on current understanding of the life cycle. And the inputs to the hydrological part are DEM data from which we derive topographic information, as well as rainfall and temperature, and the output of the hydrological component becomes the input for the other part of the model that calculates the abundance of the parasites in each of the life cycle stages, with development progress explicitly dependent on temperature and soil moisture. And the model simulates disease transmission with a daily time step at 25-meter resolution. So given these two different approaches, we were interested in understanding what difference it makes in terms of disease patterns with the aim of supporting decisions and for assessing climate change impacts on disease risk. So we show results for the River Tower catchment, which is a catchment in the UK, as an example. Firstly, our results show that accounting for soil moisture can result in different spatial risk patterns. So this is a map obtained for a particular month in our simulation period using the Ollenshaw index, so the empirical method, and at 5 kilometer resolution, which was the same as in the study that I showed before. And basically, we see that 100% of the catchment is considered at risk, and risk is higher in the north of the catchment, which is actually where rainfall levels are higher. And on the other hand, if we look at the map that we obtain using our model, we see that actually 17% of the catchment is risk-free, and there are high-risk areas, but these are scattered around the catchment. And so on one hand, the location of high-risk areas is directly driven by the rainfall pattern, and on the other hand, areas with high rainfall are not necessarily areas at high risk, depending on the antecedent moisture conditions. And the fact that with our model we actually are able to identify high-risk areas is important for disease control, as the median amount of grazable land per farm in the UK is about 2 square kilometers, so providing farmers with information at this resolution may be useful to limit exposure to infection, for example, by varying management practices and avoiding pasture at high risk in high risk periods. Secondly, our results show that adopting a daily time step, we can capture the impact of weather variability, and that this may lead to different risk dynamics. So it often happens also under current conditions that mean climate characteristics may be similar from a month to another or from a season to another, but 2 months or seasons may be different in terms of distribution of particular temperature or rainfall events. So here, for example, in our period, we have mean winter temperature in light gray and number of winter days above 10 degrees, so particularly mild days that, as I said before, are relevant for the life cycle of the parasite. And we see that on one hand, year-to-year winter mean temperature is similar, and on the other hand, the distribution of the number of these particular mild days is different from year to year. So if we take these 2 years, say, with similar mean and different distribution, and we look at the impact on disease risk using the 2 different models that I introduced before, we see that using Ollenshaw, which is in black, which is the empirical model, we get that the maximum risk of infection that is reached in the 2 years is the same, and here is about 100, and also the maximum risk of infection is reached in the same month, that is around May. And on the other hand, using our model, so health in blue, we see that a higher number of days above 10 degrees results in higher risk in this year, in 2007 compared to 2002, and the peak is reached also earlier in the season by more or less 1 month. And this also has implications for control, because knowing timing of the highest risk of infection on pasture is important for farmers to then decide when to graze their animals, let their animals graze, or when to treatment. And it is relevant in terms of assessing the impact of climate change, as in some areas it is predicted to change, things are predicted to change more in terms of variability than in averages. And so to conclude, our approach describes the biophysical mechanism underlying the transmission of fasciolosis, so it can be used to more reliably assess the impact of changing conditions on disease risk. Secondly, accounting for both climatic and environmental drivers leads to differences in disease patterns that are relevant for disease control, so we need to represent these in order to guide interventions in the future. And finally, space-time scales relevant ecologically resulted to be more meters and days compared to months, and and kilometers that are usually used, and so we need to adopt these sort of scales if we want to capture the impact of climate change in the future, including changes in variability, and if we want to support decisions for disease control. Thank you.
Thanks. We have time for a couple questions.
At the back. Well, basically the fact that there's a higher the number of days that are particularly mild in the winter is something that we know from the literature that affects the development progress of the different stages. So as I said before, there's no development of the eggs below that threshold of 10 degrees, and also snails usually come out of hibernation above that temperature. So the fact that over that season there's a particularly high number of mild days affects how quick the life cycle works, and that may lead to an earlier peak of infection in the summer.
Any more questions? Yep.
A really quick question. I may have missed something, but that's all model risk, because it's very hard to get actual cases, right?
Well, yes, we have.
So how validated is, for example, your spatial model? Zero risk.
Yeah, I haven't shown it here. We've calibrated and tested it using different data sets and also expert knowledge. So we had, for this particular catchment in the UK, we had a data set that consists of a time series of cases of fasciolosis that were detected, that were reported and at the vet labs. And so we can use those, that time series. And for space, we didn't actually have a data set for this particular catchment, but for a different one. And that data set is made of 200 farms for which we know infection, whether they were infected or not, in one moment in time, though, over a certain winter in 2015. And so these are the two data sets that we have, but we don't only use these two to calibrate and validate our model, because we have many reasons not to trust these data sets, as it often happens also with other diseases, because of misreporting, for example, or because of the fact that for the spatial data set, for example, we only have data for one particular season, whereas things may vary a lot from season to season. And so we also use a different approach that is expert-driven. So we basically formalize expert knowledge about the seasonality of those life cycle stages in the UK into a set of rules, and run our model with a large number of parameter sets, for example, and then use those rules to apply those rules to see whether we get any parameterizations, for example, that make sense. And so this is our second way of calibrating and testing it. But yeah, I didn't show this now. It's in a paper that I've just submitted. So.
Okay, thank you so much. Thanks.
Thank you.
So the next speaker is Alfi Hasan, who will be speaking about diarrheal diseases in South Asia.
Hi everyone. Today I'm going to present about my name is Alfi, and I'm going to present about understanding the hydroclimatic drivers of infectious diarrheal disease over the South Asia, and how we can project through the regional climate model. So I will start with a story. Like from the ancient time, in the region of South Asia, there's an evil spirit used to come in the villages and kill people, like village after villages. So village elders used to worship as a deity, and they used to call that Ola Bibi or Ola Devi, depending on which culture they are, and to reduce the mortality. So with the invention of the modern science, we know that the deity is nothing but a deadly diarrheal disease, cholera, and it's kind of found, we understand that around 150 years ago. But with the advancements of science up to today, is the disease problem solved? Actually, with the space science and technology, what we, in the age of this modern technology, we still have a significant amount of the mortality from the diarrheal disease. So the diarrheal disease is the second leading cause of preventable cause of death among the children under the age of 5, still today. And within the diarrheal disease, there's two diarrheal disease. One is the cholera, and another is the rotavirus. Cholera is the deadlier one, and the rotavirus is more prevalent one. So the cholera, within 100,000 people, there's 2 cases of cholera of the death, and within the diarrheal disease. And the rotavirus cases, this is the leading cause of the under 5 years child death due to the diarrhea. So if you want, we want to understand the climatic drivers, we need to first, under our epidemiology, we need to understand the transmission cycle of this disease. So it's kind of known that the diarrheal disease is transmitted through the fecal oral route, and the factors that actually act as a media is water, soil, animal, human, and foods. And all these factors can be influenced by the climate. And it's kind of known that these diseases are seasonal and also affected by the environmental factors, where environmental factor has also the climate. So climate influence, so understanding the climate influence is could be a very interesting thing to look. So usually what we had previous study had that seeing the mean climate of the effect of the disease. But things like 3-day consecutive rainfall or a heat wave for several days could actually trigger the disease outbreak. And the disease perspective can be different in the developed world and developing world. So in developed world, is there's not that much of the diarrheal disease, but when things like, let's say Houston flood, if the sanitation situation broke, then there is a possibility, and all the factors comes up, there is a possibility to have the outbreak on there. For the developing world, it's pretty much there. There's a big problem there. So there's a limited resource, and people need vaccination to cure the disease. But we need the spatial understanding of that, where we need to apply that vaccination. And in the Millennium Development Goal, we know that hygiene can be improved, that idea of the disease, the outbreak of the disease. So, but the drinking water supply, Millennium Development Goal doesn't meet that much of the, ensure the drinking water supply to the whole world. And if we want to, in the changing climate, if you want to incorporate the effect of the diarrheal disease, health issue, we need to also incorporate the climatic effect. So that's why our focus is to explore the climate extreme, as well as to see the future risk of how it could propagate. So our objective is first to establish the link between the climate extremes and diarrheal disease epidemic. Second is to have a meaningful climate extreme projections so that we can have a risk study. And the third is to have a risk, properly combining these two to have a proper risk from the baseline. So if you see the pictures, this is the top one is the cholera burden, and the bottom one is the rotavirus death. And if you see that the South Asia is kind of red all the time. So it's one of the vulnerable region. And within the South Asia, one of the climate vulnerable region is Bangladesh, where you had that all the disease, they have so the good part is, which still a good part of the Bangladesh is that it's called, it's a climate vulnerable, or bad part, and also it has a lot of outbreak each year from both disease. So that's why we selected the case study in here, and we have 6 hospitals data from 2003 to 2016. Sorry. So how we define the extremes? So we use, there's several extreme indicators that can be defined extreme of a climatic extreme event. So we selected around 46 extreme indices where daytime temperature, nighttime temperature, precipitation is considered, the lag time is considered, moving average also we considered, spatial location is considered, and we model that with each season, with 4 seasons, with a deterministic way, and so also the randomness in a statistical way. So after that, this is kind of the result or the cycle of the disease. So the left part is the rotavirus, where you see there's a winter outbreak. Usually it happens, and in the monsoon there's a little outbreak. And the right side is the cholera, which is actually opposite kind of the rotavirus. That's the cholera is the bacterial disease, and the rotavirus is a virus disease. So they act differently in outbreak. And the cholera happen in the spring and the fall. So it's a dual peak nature in there. So this is a spatial picture of that with our hospital surveillance. So I just want to explain that how the situation of the country. So to correlate that, what the relationship we have in the future, like in the next life. So in the top part of the country usually warm, and the bottom part is usually cold. And if you see the west part usually the dry, and the east part is usually the wet. So that sort of thing actually plays to propagate the disease. So this is our relationship with the climate extremes. So what we found that from our analysis so winter, it's known the rotavirus is more transmissible when the environment is dry. So that means if there is less number of rainy day in the winter, or number of rainy days in the winter, that actually help the virus to propagate in that. And it's also require certain nighttime temperature. So colder temperature is better, but it's not always, it's not high correlated, but the certain temperature in 16 to 21 degree, we found that that's actually correlate. If you see the figures in the bottom, there's two variables actually correlate or together. For the cholera cases, the significance was in the fall one, where we know that like fecal oral route always connected with the water. So that means flood actually, so we found that the indicator that is RR5, which is 5-day consecutive rainfall. If there is any 5-day consecutive rainfall in any places, it's high probability to have a flood or waterlogging, whatever it is. So there's a high chance to connect the fecal oral route of the transmission cycle. So that's why in the fall there's a high correlation with the cholera and with the RR5, the indicator of the extreme rainfall. So this is our model from the rotavirus. So if you see the observed one is the green one, and there's a deterministic part where we try to explain with the physical processes and that, like try to at least have the deterministic part in there, which is the dot line in there, and the rest of the randomness we applied the statistical model like Sarima model, and then we adjusted out the whole thing. So we did modeling in the 4 separate season and then combine it in there. And now we know the extreme is affecting the disease. How we can project that? So there's a problem that like projecting the extreme, we need to project this more meaningfully. So to do that, we created an observed gridded data. We applied the dynamic redundancy cortex domain, and we applied the RCP scenario. We applied the bias correction. All these things. I mean, why we need the of the model and derived, and why need the extreme corrections, we can see in these figures. So in the left side, if you see that this is observed data, and if you see the top 5 figure of 5 model, it doesn't actually capture. This is the number of the rainy days. It doesn't capture well with the observed data in the baseline period, what you observe. So the thing is that if you project this in the future, it will give you a wrong idea actually how the extreme is. So we need to first at least correct that and then project it in the future. So that's the same thing we found that in there, like the top one is the bias corrected result projections, and the bottom one is uncorrected projections. So you have two different intuitions from the same projections. So yeah, we are corrected the extremes because we are analyzing with the extreme. We understand the relationship between the extreme of and the disease, and then we combine it and see the projection change of the disease outbreak in the future. So this is the percentage change from the baseline of the disease risk. So if you see that the southern part, as I recall, that the country is much drier. Country is much colder in the southern part. So the extremity of colder is increasing in the models. That's why the risk is increasing in there. In the same situation, this is the left part is the mid of the 21st century. The right one is the end of the 21st century. So this is the two. So the extremity is propagating in the future. And the second one is the cholera. So the cholera is there you see there's negative in there, like there's some disease risk is reducing some cases. So why is it happening? So as I recall that, like in the fall, we said that the RR5, the extreme rainfall, is one of the indicators that actually affecting the disease. That indicator in the climate models projections is so uncertain. So some models shows is getting higher, some models shows lower. So the uncertainty kind of result in the mean models result, that is negativity and positivity. But eventually, if we see the average from the left figure to the right figure, this is the mid of the 21st century, and this is the end of the 21st century. It's kind of increasing in the future as well. So conclusions. We just look at the climate influence of the disease in here. So we actually use the Z factor, not the incident. So we kind of remove the effect of try to remove the effect of the population. This is one of the reasons we use the RCP scenarios. RCP scenarios is not an emission scenarios, it's trajectories. So it's only considered the greenhouse emission backwardly. And then our model could be, this is another constraint with the data availability. Yes, it's 2003 to 2012, 2016, but ideally climate would be 30 years. So if we have more data, we can validate more accurately. And then, yeah, other regions could be useful if we have enough data. If we find the Indian data or African data, that would be useful. And with the integration of the satellite image, the relationship could be improved in future as well. Thank you very much for your attention.
So we have time for one very quick question. Okay, we'll move on to the next. Thank you so much.
So the next speaker is John Balkus will be presenting, and climate-sensitive infectious diseases.
Good afternoon, everybody. It's always an honor to be the last speaker in the almost last session of the day. Okay, I'm sorry, I'm looking at this, which is, okay. Great. So I'm not Juli Trtanj, who was supposed to give this talk. Juli was unable to make it, but I'm presenting this on behalf of Juli, as well as other colleagues with the GCRP, Mark Shimamoto, who sadly has left us and come over to the AGU, and also Claudia Brown. And it's fitting that I go last today because I think a lot of what I'm going to present to you, which is the outcome of an interagency process looking at the use of climate models and climate information for predicting and forecasting outbreaks of infectious diseases, a lot of the conclusions we came to in this process are very consonant with the 7 talks that we've had already. So there are many different emerging infectious diseases. I'm not going to spend a lot of time on this since I've lost a little bit of time. But these are both of interest in the United States. I'm sorry. I'm having problems with sure why that happened. In the United States and also in areas where the United States has interests. The genesis of the federal science plan that we were working on actually came from the previous administration's efforts on climate change and national security. And so we were bringing together not only the interests of the climate change science community, but also the international development community, the Department of Defense, and the intelligence community. We had a sense that there were many different programs in the United States that were either conducting research or actually trying to answer questions related to forecasting infectious diseases, but as often happens, they were not well connected and not well integrated. So part of what we were trying to do was to pull these together to define what were the critical science questions. And what happened was that we kind of morphed from a primary research agenda into looking at how we can start to pilot and operationalize and integrate the different efforts to actually do these forecasts and see if they can actually add value to the different government programs. Juli made sure that I included this slide, which just reiterates for all that the work that we do in the climate change research program is actually congressionally mandated and maintains that congressional mandate from the Global Change Research Act of 1990, which specifically directs the Global Change Research Program to not only understand changes in climate, but to assess, forecast, and assist the country in dealing with the impacts of climate change. And the work that we're doing is actually being coordinated by an interagency group on climate change and human health that Juli and I are two of the co-chairs of, along with George Luber of the CDC. And this slide just describes our mandate. I, again, won't spend a lot of time going over that. So the goal that we have, and this is something that we have all been talking about today, is trying to transition infectious disease control from being purely responsive, from relying on systems of surveillance and disease detection, to being able to use climate and environmental information on the drivers of the infectious diseases to get ahead of the game, to be able to forecast both areas and timeframes that will be of higher risk. Depending on the actual disease that you're talking about, that will determine different kinds of interventions. For malaria, for example, it may be pre-positioning medications in areas that may become inaccessible because of rains. For something like dengue, it may be household interventions for controlling vector sites. So it's very hard to generalize, but in general, what we're trying to do is to give forecast and lead time to public health officials in order to be able to prevent outbreaks before they start being detectable in the human population. So I think this is just a building slide to show what usually happens, which is the response doesn't get generated until you have several laboratory confirmed cases, and if you can get ahead of the game, you have the opportunity to mitigate the peak of that epidemic curve. In pulling our information together, we were looking for areas where this has actually been operationalized. We've had some great presentations about research programs, and I'm very eager to have some conversations about where this information is actually being utilized by public health officials. This is an issue that we face. The prime example that we found from our work where this is actually functional and leading to interventions that are preventing disease outbreaks is actually Rift Valley fever in Africa, where collaboration among NASA, USAID, NSF, et cetera, and the CDC have been able to develop an operational seasonal forecasting system that gives information to farmers and allows them to take interventions months in advance of the outbreak. So as I mentioned, the motivation of this was recommendations from the Climate and National Security Working Group that was formed under the previous administration to create a comprehensive climate and health science plan. That plan had several different components. What we wanted to start with was actually this project on looking at forecasting infectious diseases. There was also a recommendation to look at the predictive modeling and failure analysis for healthcare facilities and extreme events, and I talked about that in the session a couple of hours ago. We're also considering doing a focused look at air quality and looking at combined impacts of wildfires and other aeroallergens in relationship to climate change. Ultimately, our hope is that we take these pieces and operationalize them in a kind of topic-specific way, but also come up with a prioritized comprehensive science plan for the myriad of impacts of climate change on health. And we're hoping to do that in the coming year. So the process that we went about was to have 2 open webinars where we brought in representatives from inside and outside the federal government. The first was to try to capture the state of the science, so that was really trying to bring in the academics who are doing the research. Some of you are in the room. The second webinar was to bring in the end users of the information, so the public health departments, the development agencies, et cetera, the military, the intelligence community, to understand their needs, to understand how they're using this, whatever information they're getting now, and then what they would like to see out of a science plan. And then we held just a federal interagency workshop, which again started off being something that we thought would lead to a research agenda, but actually morphed into something that was much more operational in orientation, looking at what are the existing programs in the different agencies that could use this information, and how can we work together to start to pilot at least the use of climate forecasts at the subseasonal and the seasonal level into the work that they're doing. So I'm going to summarize the findings that we had from our web conferences. We had the state of the science, where we had people telling us that we actually have, it's not a question in most cases of understanding a modeling approach or having models that can provide the forecast. It's much more, as we've heard, much more often a question of availability of data, whether that's the entomologic data in the case of vector-borne diseases, whether it's good surveillance data, especially in the case of waterborne diseases and vector-borne diseases, but also the availability of downscaled seasonal and subseasonal climate and weather forecasts for the parameters that are most important. As we've noted, sometimes it's very hard to get the seasonal, subseasonal forecasts of precipitation in a way that can drive the models in an accurate way. One of the points that emerged was, from a research standpoint, even being able to retrospectively recreate data sets, if there are ways to go back and, for example, go into hospital records and not rely on the reporting and surveillance systems, but go back to other sources of information and recreate these data sets, would be very valuable to develop and validate the models. Some other kind of specific points that emerged from our state of the science were some advances in methods of predictive modeling. We had presentations about ecological niche modeling, where the disease was actually treated as if it were a vector species itself. So that in modeling the disease transmission, you are actually integrating both the vector biology and the aspects of transmission to humans in order to kind of shortcut your way around developing a fully mechanistic model to have all the full parameterization and be able to predict in that way. So this is one technique that people were using. It also became clear that the Department of Defense and intelligence communities both had, the military had unique capabilities in terms of their patient populations spread around the world, for which they had both public health control, but also much better surveillance than most other health systems. The intelligence community, we found, was much more of a passive observer of the forecasts and models. They were not actually doing the modeling themselves. Lastly, in the United States at least, we found limited uptake from the public health community for a number of reasons, largely low capacity, cutting budgets, and a sense that given the resources they had, that the value added from the uptake of forecasts and predictive models just wasn't worth the effort to create and generate that kind of information themselves. And I think there's a value proposition for a lot of the public health departments in the United States. So another thing that I'm anxious to hear how that uptake is going for the others. Again, I mentioned Rift Valley fever being a model of uptake. From the users conference, we had these examples of places and diseases where there were some pilots going on in different places. And again, from Jeff and others, we've heard of some of the cases where public health departments are starting to pilot this. And I just want to, the bottom point there, I haven't heard much recently, but legislation passed by the Congress specifically was calling for the use of seasonal and subseasonal forecasts to be used for societal needs. So there was, again, a congressional mandate to be looking at some of this work. Again, in the workshop summary, we got into a discussion of implementation more than research agendas, and so we focused a lot on some of the barriers and challenges. From a scientific knowledge standpoint, we had the sense that there were many different groups, different research agencies and research institutions around the world that were piloting this, but there wasn't a home, there wasn't a central center that was doing a lot of coordination among the different centers. I can get into more detail if you have questions about that, but there was a real need for this kind of integration across at least the US government agencies doing this work. There was an issue about having an authoritative forecast and who would be the agency that would take the risk, if you will, of producing a forecast that would have some uncertainty and have the liability of those forecasts being correct or not. Again, the idea, the need of a central, more authoritative source. And then lastly, this idea that there's a little chicken and egg. It's hard to get the resources to develop and refine these systems in a skeptical public health population where they don't have the value proposition proven, and so getting the funds to it's hard to get the funds for research to develop it when the uptake isn't guaranteed. So I'm going to run out of time here. We came up with a lot of different next steps. Some of the more exciting ones were working with the Agency for International Development, which is already starting to implement a mandate to look at climate risks to their development investments. So they're starting to look at risk mapping on a global scale to figure out where and among the countries that they're working with they are at most risk of infectious diseases threatening the health systems. We're also looking in collaboration with groups like NIHIS and looking at heat to start looking at the seasonal and subseasonal forecasts and try to start translating them for health implications in the United States and also in different parts of the world. I just want to close by mentioning two funding opportunities that are relevant to this. And again, this is a slide that I did not make, but if you look towards the second and third bullets there, NOAA is producing a funding opportunity specifically on climate-sensitive health risks. This is hot off the presses. There's going to be a webinar, as you can see, tomorrow. So all of you in the research community, please take a look at this. And then there's also a funding opportunity through the NIH. The MIDAS program has been in existence for a long time. It has tended not to really focus on environmental drivers, but they are redefining the MIDAS program. There is a current funding opportunity there. There is more openness to looking at climate environmental drivers, so I urge researchers to look at that funding opportunity as well. There's our email addresses, and with that, I'll close. Thanks very much.
So we're out of time for questions, unfortunately, but do feel free to come ask John some questions afterwards. This is the end of the session. I want to say a big thanks for all the speakers, and yes, do check us out online afterwards. The session will be available. Thank you.